Good afternoon, everyone. Today I will be presenting to you my presentation over the transcription factor bHLH92 and its effects on monoterpenoid indole alkaloid biosynthesis, possibly via a protein-protein interaction in Catharanthus roseus. When a plant is attacked by a feeding insect, the regurgitant produced by the feeding insect elicits a defense response in the plant. Local wounding, such as like this wound right here, by the herbivore, leads to a burst in newly synthesized jasmonate. This newly synthesized jasmonate triggers a biosynthesis of specialized metabolites for defense throughout the plant. One such example of this metabolite is monoterpene indole alkaloids. Monoterpene indole alkaloids are vinca alkaloids derived from the indole and iridoid biosynthesis pathway of the jasmonate response signaling pathway. It is used for defense in Catharanthus roseus. Two MIAs of importance are vincristine and vinblastine. They're derived from the condensation of tryptamine and cyclogen, which are products of the indole and iridoid pathways. Vincristine and vinblastine are vitally important components in the treatment of certain cancers. Vincristine and vinblastine are actinioplastic agents. This means they destroy, inhibit, or prevent growth and/or spread of tumors. For example, leukemia is characterized by an elevation in white blood cell count. When patients are treated with vincristine and vinblastine, it lowers said white blood cell count in said patients. But there's a problem with using vincristine and vinblastine. It's expensive and in limited supply. This is due to the fact that the plant produces these MIAs in low yield. So it takes about 500 kilograms of leaf biomass, of Catharanthus leaf biomass, to produce just 1 gram of product. And it's also impractical to produce these or chemically synthesize these in a lab setting in a large scale program due to the structural complexity of these compounds. Thus, metabolic engineering strategies have been employed. But they've come across a problem. You can't just upregulate a single transcription factor or their downstream targets. It doesn't lead to an increase in MIA accumulation, net MIA accumulation. But instead, there's a large regulatory feedback loop that's controlled by a plethora of JA-responsive transcription factors that either activate or repress known points throughout the biosynthesis pathway. There are many different family groups of transcription factors involved in this, and with members that activate and some repressing. What this should symbolize to the viewer right now is just how complex the sets of knobs and levers and switches that need to be affected to actually cause a downstream increase in MIA accumulation. One important family of transcription factors are the basic helix-loop-helix transcription factors. This protein family is characterized by its structural motif. It has two distinct regions. The first region being the basic region, which is involved in DNA binding. So as you can see here by the diagram, the basic domain is able to bind to the DNA sequence of its target gene via a consensus sequence called an E box or an enhancer box. One example of an enhancer box is the G box. The G box are commonly present in promoters of MIA biosynthesis regulators, which is important for the audience to know as we go into this presentation. Second is the second distinct region, which is the helix-loop-helix region, which allows for functional dimerization, allows to form heterodimers, which allows for the ability or allows the formation of a large number of potential DNA binding complexes with different biochemical properties, including DNA binding, activation, or repression potentials. This thereby creates a multitude of opportunities for bHLH transcription factors to regulate different genes and sets of genes. Our lab has invested a lot of time and effort looking into basic helix-loop-helix transcription factors in Catharanthus roseus in the MIA biosynthesis pathway. One well-acknowledged master regulator, MYC2, has led to the identification of the repressor MYC2 target 1 bHLH transcription factor. An important bit of information is that these GE boxes are present in all the MIA gene promoters, or a lot of the MIA gene promoters, suggesting that there are additional bHLH factors involved, as can be seen here. Our MYC2 master regulator might be activating gene expression, while repressor of MYC2 targets represses said target genes. So you can start to see the formation of the beginnings of like a regulatory feedback loop there, which leads us to the identification of another potential regulator, bHLH92, which is tightly co-expressed, as can be seen here from the co-expression analysis done in our 2018 paper, that this could be an interesting target for regulation. bHLH92 is in subgroup 2 and is homologous to the Arabidopsis bHLH91. We ran a co-expression analysis to see if bHLH92 was co-expressed with MIA pathway genes and regulatory genes of the MIA biosynthesis pathway. And as you can see here by the red, that is true. There is a significant density of that amount of co-expression going on between bHLH92 and MIA biosynthetic genes. Therefore, we hypothesize that the transcription factor bHLH92 is a transcription regulator of MIA biosynthesis. Therefore, we seek to elucidate the role of bHLH92 and its possible combinatorial regulation of MIA biosynthesis. For our experimental plan, we did the co-expression analysis to identify target candidate genes. Next, we'll focus on bHLH92 by functionally characterizing bHLH92 using floral infiltration for overexpression, hairy root transformation, and virus-induced gene silencing. From there, once we functionally characterize our bHLH92, we will then look into how the properties of the promoter binding transactivation of bHLH92 using a protoplast transactivation assay. From there, we will then investigate possible combinatorial regulatory genes using a toolbox of different methods and protocols, as can be seen in the red, to answer said question. For our floral infiltration experiment for bHLH92, we generated 4 overexpression lines. Of these 4 overexpression lines, we selected overexpression line 2 and 4 as they were kind of a standardized median or middle example to compare against our EV control. For the expression of AP2-ERF transcription factors ORCAs 2, 3, 5, and 6, we saw an increase in expression levels. For the genes in the indole pathway, as well as the master regulator MYC2, we saw an increase in TC expression. And for the iridoid pathway, we saw an expression increase for 6 different iridoid pathway genes, which led to the proposed model, as you can see here on the right side of this PowerPoint, that bHLH92, after JA, a burst of JA occurs, the expression of bHLH92 leads to an increase, or leads to the regulation of the transcription of downstream transcription factors, in this case ORCAs, which then regulate downstream expression of target genes within the MIA biosynthetic pathway. For our virus-induced gene silencing, we generated knockdown lines for gene of interest using VIGS, which is a reverse genetics approach. We use the PDS and CHLH gene for visual markers to identify where the virus kind of infiltrates to, where we should harvest from. So where would our transformation, where would our virus-induced gene silencing have occurred? And we use PDS and CHLH to, via its photobleaching phenotype, to identify where we need to extract from. We are currently in the process of measuring the alkaloid content from these transformants. For the hairy root transformation, the reason we use the hairy root transformation is because there are no standard protocols for generating stable transgenic lines of Catharanthus currently available. Thus, we have to use the hairy root transformation method. Currently, I'm in the process of generating hairy root transformants of overexpression lines, which have a substantial increase in expression levels of bHLH92.
You're at 10 minutes.
As well as generating RNA interference lines, which knock down the expression levels of our bHLH92. Once we've generated transformants, we will then excise the portion of the root that's the visible hairy root that's visually distinguishable from the rest of the plant. We'll excise that and place it on a selection media, as can be seen here. The selection media will have a specific selection antibiotic. If it's able to grow and proliferate, then we will then put it onto its own plate. From there, I will excise a small portion of the top to actually verify on top of the selection media. So the selection media is 1 degree of verification, and then the second degree of verification would be using the gust staining to verify our transformant has the proper insert. If it does, when exposed to X-fluke, it will have the blue phenotype, as you can see here. From there, after we verified, we will then use an RNA expression analysis to see the fluctuations in expression levels in our hairy root transformants. And on top of that, I will continually propagate roots for future experiments analysis. For my protoplast transactivation assay, we first fused the Gol4 binding domain to the bHLH92 to see what regulatory function does bHLH92. When these are fused, Gol4 binding domain will attach itself to its respective response element here above the cauliflower mosaic virus 35S minimal promoter, which is just like a Tata box. The Gol4 binding domain fused with the bHLH92, the Gol4 binding domain will bind to the response element, and the bHLH92 will either activate or repress the activity of the luciferase, downstream luciferase open-reading frame gene. From the results, we show that bHLH92 is an activator. Then we did a second protoplast transactivation assay to see if bHLH92 actually directly transactivates TDC or IS, which are the genes that we saw earlier in the floral infiltration that were like increased when bHLH92 was overexpressed. They increased in expression levels. So we know that they interact, there's something there. And so we wanted to see if bHLH92 directly binds to TDC and IS promoter, which is fused to a luciferase open reading frame. If it's able to bind, then it will transactivate and increase in the luciferase activity as seen in the first one, but that was not the case. It was unable to transactivate in tobacco protoplast. We believe that it's an interesting question because why was bHLH92 able to transactivate or was able to increase the RNA expression of target genes in the floral infiltration overexpression lines, but was unable to transactivate the TDC-IS promoters in our protoplast transactivation assay? And that leads us to the speculation that maybe there's a co-activator involved. Maybe a partner is needed from Catharanthus for this activation to occur. Therefore, we looked to the literature to find an answer for that, and we found that in QE 2016 that they reported in-plant interactions of the Arabidopsis subgroup 3 bHLH transcription factor or co-activator dysfunctional tapedum-1, that it interacted with the member subgroup 2 bHLH91 in Arabidopsis. bHLH91 is an activator and does not bind to the promoter, but rather interacts with group 3 to actually activate its target genes. bHLH91 plays a role in nuclear localization of DYT1. The closest homologue of bHLH92 in Arabidopsis is the subgroup 2 bHLH91. Currently, there are no known subgroup 2 bHLH transcription vectors known to regulate specialized metabolism in any plant currently. Therefore, with the DYT in mind, we identified a bHLH of subgroup 3 homologous to DYT1.
You've got about a minute left.
With my with my time, I will focus on its function to identify its function, and then to see how it co-regulates with bHLH92. As you can see here from the co-expression analysis data, that it does interact with a ton of the MIA regulatory genes throughout the MIA biosynthesis pathway. Thus, I made a new updated version of the proposed model with DYT1 needed for the co-activation of downstream target genes. In summary, Catharanthus is a valuable medicinal plant. MIA transcriptional regulation network not fully elucidated. We hypothesize that there's additional bHLH transcription factors that activate activators present in our regulation network. We've identified subgroup 2 bHLH92 and speculate subgroup 2 bHLH92 interacts with a subgroup 3 bHLH-DYT1. The significance of this paper is that somehow Catharanthus has somehow possibly evolved a co-regulatory mechanism of subgroup 2 and 3 bHLH transcription factors for specialized metabolite biosynthesis. That is the end of my presentation. Thank you for your time. Are there any questions?